All right, so in Chapter 10, we're covering mitosis, which is, didn't mean to do that, cell division. The whole purpose of the process I'm going to explain today, but you need to understand that you yourself are undergoing mitosis continuously. Mitosis explains a lot of the phenomena that a lot of people they just take for granted, and I'm going to explain those today. But continuously, you're going through mitosis. It's a non-stop process. Big picture, though, all we're doing is dividing cells. We're cloning and dividing. Cloning and dividing. So that's what we're going to be looking at here. When we say mitosis, the bottom line is, the purpose behind it is for growth and maintenance. And I'm even going to throw the word in there, repair. And I'm going to, of course, explain and justify my answer there. But the reason why cells clone themselves and then divide is so that you can grow, so that you can maintain what you have, or so that you can repair what you've damaged. This only occurs, mitosis only occurs in multicellular eukaryotes. What does that mean? Multicellular, and I don't want to say it only occurs, but let's talk about what type of cells undergo it. So it occurs in multicellular eukaryotes. What does that mean? What does multicellular mean? You have many cells. You have more than one. You're not unicellular, you're multicellular. What does eukaryotic mean again? It's got a nucleus. It's got a true nucleus. There are only 4 kingdoms that are eukaryotic. What are they? Protists, animalia, fungi, and plant. Those 4 kingdoms, those organisms undergo mitosis. And I'm saying that now because we'll come back to it in a little while. Bacteria don't go through mitosis. Only eukaryotes. It mentions here that mitosis is used for asexual reproduction in single-celled organisms. Still eukaryotes, though. What does asexual mean? How many parents do you have? You have 2, yes. But in asexual, how many would you have? 1. So if I'm making a baby on my own, my baby's going to look just like me, which is the whole concept behind clone and divide. Clone and divide. Okay? Now, when you were born, roughly what size were you? Okay? So you can say like this. Let's say about how much do you think you weighed? 7 or 8? Okay. So I'm just going to say that I was 8. I was something. I mean, obviously, because I'm still here. But I think I was like 8 something. But how did I go from being 8 pounds to 140? Mitosis. Bottom line. When your aunt comes and pinches your cheeks or your grandma, and she's like, how did you get so big? Mitosis. Duh. That's how you grow. The only reason that you grow is because of mitosis. Because if you were born this size, your mom is probably not with us any longer. Okay? So you grew. So that's what I mean by growth. Now, maintenance. Do you continue to grow in size your whole life? No. You eventually plateau, or else we would be humongous. In order for me to stay this size, I have to replace the cells that die or can't be used anymore. For example, my skin cells, how many of them am I losing right now? Thousands. Thousands and thousands every minute. But if I didn't make new ones, what would happen? I would have no skin. So you would just see like my fascia, my muscle, and my adipose tissue. You would see all that, because it wasn't being replaced. So when I say maintain, we have to replace the cells that die or fall off or get damaged or whatever. we have to replace those. So that's what I mean by maintenance. And I said I want to throw in repair there. Because if, like for example, earlier this week, I got my hand caught in something and it kind of bruised a little bit and it tore the skin. But is it going to stay like this forever? No. Because of mitosis, new cells are going to be created and they're going to replace these ones that are damaged. And in about a week, it's not going to be there anymore. So that's what I mean by repair. Mitosis is used for growth in order to maintain what you have, and then repair of anything that's been damaged or broken or so on. You break a bone, that healing process, mitosis.
I have a question.
Proceed.
You heard of the people with like, I think it's called like butterfly skin? And it's like when they have like a bunch of like blisters all over their skin.
Okay.
They can't. Like they basically stay like that forever. Like there's no cure for that. Is it that they don't go?
Okay. First of all, if it's blisters continuously, there has to be something genetic or there's something in their environment that they're exposed to that's causing that. Those would be I don't know what condition you're talking about. Okay. So my first and foremost answer would be I would love to research it. But just based on what you're saying, if it's continuous, either something in their environment is assaulting them regularly, like there's an allergy or something, or there's something genetic. And if it's something genetic, we can't fix it. So it's not that they can't conduct mitosis, because if they didn't, their skin still, do you see what I'm saying here? Like, now, it's obviously not healing, which makes me think that if it's not the environment, it has to be genetic. So I would need to and look into that more. Like, I hear about diseases all the time, and I don't want to be like, oh, well, it's this. I like to know what I'm talking about, so I don't want to speak ill-knowledgeably. But if I had to hypothesize, that would be my thoughts there. Good question. So at the end of the day, after my cells have gone through mitosis, my result is 2 new genetically identical daughter cells. So they're clones. What do I mean by genetically identical? They have the same DNA. They have the same DNA. And that makes sense because I want them to replace an old cell. So if I'm making skin, I want my new skin cell to look just like this one. I don't want my skin to change any. Okay? So I like to use diagrams that aren't specifically in your text. I'm going to turn off a light and see if it doesn't do more contrast. So I have a diagram here of a eukaryotic cell, and I know it's eukaryotic because it has a nuclei and it has some membrane-bound organelles. Okay? And then this one cell duplicated its organelles, and then it divided. So once this cell divides, is this actually here any longer? No. So it was a parent cell, and it divided into 2 daughter cells. Before I move on, why is it daughter and not son cells? Okay, so I feel like first of all, well, I'll say this last of all. First of all, in all science, it will always be mother, daughter, sister. It will always be female-oriented when it comes to reproduction because, generally speaking, in the reproductive process, the female usually conducts the majority of the activities. I also believe that this is science's way of making it completely known that females are indeed superior to males. No, maybe not. That was my last of all thought. So it will never say father cells or son cells or brother chromosome. No, it's not. It's all sisters. Okay, so just so you know, especially those of you going further on into science. So that's actually something that will be consistent, but I did want to address that there. So in mitotic cell division, this is my summary slide. I have to start off with DNA replication. So what is happening during DNA replication? I'm making a copy of my DNA. Like it's really, I'm just replicating my DNA. Real quick question. Which cells in your body have DNA? Mitochondria? What'd you say? Which cells in our body have DNA? All of your cells. All of your cells. Okay. Surprise. All right. Extension on that real quick question. If all of your cells have DNA, which they do, fact, which cells have the most? Parent cells. So how do you know which ones are parent cells? Okay. The one with the most DNA. Fantastic. Additional hypotheses? Okay. So if I had to say a type of cell in your body, and you have, let's say, over 200 types of cells, which cells have the most DNA? Do they all have the same amount of DNA? They all have the same amount. They all have the exact same amount. Yes. Can you see where I'm going with this? Now, okay, one more question. If they all have DNA, and they all have the exact same DNA, same amount, how on earth do these cells look like skin and these cells look like eyes? Okay, that's the correct answer, but what does that mean? She said proteins. We got to backpedal a few steps. I heard someone say shape. So they do have a specific shape, which means that they have a specific function. That's all true. But if we all have the same message, so DNA is a message, it's an informational molecule. Every cell has the same one. Why do these ones look like skin and these eyes if it's the same message? What do we call the sections of a message? A segment of DNA is called a gene. Yes. I can take your skin cell, one skin cell. You don't even have to be around. I can take it off a Coke can. And I can get the DNA for your liver, for your kidneys, for your blood, for your everything is in there. It's just that because it's in your skin and it looks like skin, the skin gene is turned on, but all the other genes are turned off. Your genes only have two options. They can either be on or off. And I'm going to take a little bit further because Andrea did mention this. If that gene is turned on, it makes proteins. So if a gene is turned on, it makes proteins. So in my eyes, my eye gene is turned on, but my heart gene is turned off. My kidney gene turned off. Skin gene off. So I'm making eye proteins, which is why my eye is the eye. So the very first thing I do is replicate that DNA because I want to make an identical copy of that cell. And then the second overall step is that I want to segregate or separate. And I put both words because I want you to know they're the same thing. But I want to evenly separate that genetic material. Why evenly separate? So it's balanced. Well, I know she said balanced. What did you say? I didn't. Yeah, because I want it to be identical, the same. So I'm going to simplify this. I realize we have 46 chromosomes, but let's say that I have a cell that has 4 chromosomes. When I replicate that, how many chromosomes will I have? So I have 4. I replicate it. I have 4 more, so I have 8. And if they divide evenly, I should have. If they divide evenly, I'll have 4 in each of my new cells. So I started off with the parent cell with 4. I replicated the DNA, so now I have 8. Then it will divide, and each new cell will have 4. So identical to that parent cell. Now, the reason we have to have an equal separation is because if I have those 8, which is what I had after the replication, what would happen if 5 went to one and 3 went to the other? There would be dysfunction. And I feel like that's a very vague answer, but it's correct. This is when mutations occur. We have diseases, conditions, and so on. And that's actually where we're going to get to later on in this lecture. So we want that equal separation so that the cell functions the way it's supposed to. It has all the material it needs to function correctly. Again, at the end, you'll have 2 new but genetically identical daughter cells. So again, this parent cell gets larger as it replicates that genetic material, and then it itself will split. Will this parent cell still be a thing? No, it won't. It has literally split into 2. Okay, so some terms I want to make sure you're solid on. First of all, chromosomes. And we know that in actuality, chromosomes are just your DNA. They're your genes are your DNA. DNA all wound up and organized is your chromosome. Do you remember what shape our chromosomes are? Linear. We have linear chromosomes. Eukaryotes, like you and myself, our chromosomes are a linear shape, a line. The number of chromosomes you have specifies what species you are. So we have 46, and every organism that has 46 is considered a human. Let's say that cats have 12, so everything that has 12 is a cat. You can only reproduce with an organism, and what I mean by reproduce is you can only successfully have children with an organism that has the same amount of chromosomes as you. So you may have intercourse with something else, but you can only have a baby within your species. Your question?
So if you said, for example, you said that there's like 12...
Let's just say, yeah.
...in a cat, does that include all felines? Or just...
No, that was just an example I just pulled out of the air.
No, I know.
Oh, but no.
Include...
Are you talking about this specific, like...
So like a tiger, a lion...
No, ma'am. No, because a tiger is a different species than a lion.
Okay.
Now, if their chromosome numbers are close enough together, which is the case with lions and tigers, which is how they can reproduce and make a liger. So let's say one may have 12 and one may have 13. It would be super, super close. I truly don't know the number of chromosomes for those two.
No, I know,
but if they could successfully reproduce, they have to be close enough that that baby's viable. But that baby itself cannot have babies.
Okay.
So, it's a very good question. If I knew the numbers, I would rattle them off for you.
No,
but,
I know. Yeah,
but
it
yes.
was just for the...
Just to get the concept.
Yeah.
Where will we find our linear chromosomes? In a nucleus, because we're a eukaryotic cell. So I have this cell here, and I kind of highlighted this big nucleus, and so you can see the different chromosomes in there. All right? Now, the term diploid and haploid, do you know how many sets of chromosomes you have? We have 23 pair. That is correct. How many sets do we have? 2. How did we get 2 sets of chromosomes? 1 from your mama, 1 from your daddy. So daddy gave you 1 set, mom gave you 1 set, and as a result, you have 2 sets. So organisms that have 2 sets of chromosomes, like you and I, we refer to them as diploid, which means they have 2 sets of chromosomes. In fact, excuse me, every cell in your body is diploid with the exception of your gametes. And if you're a female, your gamete is an egg. And if you're a male, your gamete is a sperm. And in that case, your gametes only have 1 set of chromosome. They are haploid. Why do your gametes only have 1 set and not 2? Because mom's gamete has to meet dad's gamete to make you. And if dad's gamete had 46 chromosomes and mom's gamete had 46 chromosomes, when it came together to make you, you would have 92. You would be miscarried. In fact, the majority of miscarriages are a result of a chromosomal misnomer, meaning that there's either too much DNA or too little DNA. And as a result, you are selected against in utero because your body cannot survive outside. You have to have 46 chromosomes. You could potentially have 47 and survive, but you would have complications. You could potentially have 45 and survive with complications, but you couldn't have 40 and make it, and you couldn't have 50 and make it. Does that make sense? You have to have 46 because each chromosome has a gene, multiple genes, and each gene makes a protein. So when you duplicate proteins, it's too much of a good thing, and then it becomes bad. But that's also a whole different lecture that we'll go into for the next test. So everybody's solid on what chromosomes are. All right? Next term. And these aren't in the basic eCampus notes because I added them because I saw a lot of people were getting confused. So, the topic is DNA replication. But the term is homologs or homologous chromosomes. Same thing. And then sister chromatids or chromatids, depending on where you put the emphasis. So let me tell you what I'm dealing with here. This image right here is what we call a karyotype. And for those of you who go into genetics, you're going to analyze these, and they're so much fun. But how I get this image is you extract a cell, and from that cell you take out the nucleus, and from the nucleus you take out the DNA. And in the old days, we used to take a picture of the DNA using a very special camera, of course. And it's not organized like this, so we would cut it out and then organize it like this. Now we have computers that can do all this for us, and like that fast. But let me tell you how I have this organized. These are all 46 chromosomes, and these chromosomes happen to belong to a healthy male. They are in order from the largest chromosome to the smallest chromosome. They're in order from the largest to the smallest chromosome. And there's 2 of each chromosome. Why do you think there are 2? Why do you have 2 of each chromosome? 1 from your mama, 1 from your daddy. And this very last set right here determines if you're a boy or a girl. So here I have a boy. Now, in my karyotype, when I've organized all of my chromosomes together, chromosome 1 from mom, chromosome 1 from dad, chromosome 2 from mom, chromosome 2 from dad, 3 from mom, 3 from dad, and so on, which is why you have 2 copies. Because these are the same chromosome, but they came from a different source, we call them homologs, or homologous chromosomes. So they're the same chromosome, they're chromosome number 1, but they came from a different source, 1 from mom, 1 from dad. Homologous chromosomes. And you have this set up in every cell in your body, but except for your mature egg and sperm. Now, these homologs are solid, but when it comes to DNA replication, which is what they have to undergo in order for mitosis to occur, if you have 46 chromosomes, after the DNA is replicated, how many will you have? If you have 46, and we make a copy of that, how many do you have now? 92. Because every chromosome has to be copied, true or false? True. We don't want to miss out, because then we have an issue. Bless you. So, what I have here is, let's say, I have my homologs for chromosome 1. So I have chromosome 1 from mom, chromosome 1 from dad. Those are homologs. After DNA replication occurs, because you have to make a copy of both, I have my original from mom, my copy from mom. My original from dad, my copy from dad. These are now called sister chromatids. because they're an identical copy of the same chromosome. They're an identical copy of that chromosome. Collectively, these are still homologous because they're still both chromosome 1. It's just that I made a copy of them. So the copies are called sister chromatids. Copies are called sister chromatids. Here I just have my homologs, my homologous chromosomes. But once DNA replication occurs, you have 2 of each one. So they're called sister chromatids. Anybody confused on this terminology? Okay. So the whole focus of this chapter is the cell cycle. In high school, we just taught you mitosis. So it was prophase, metaphase, anaphase, telophase. I choose to teach it the way I'm going to teach it to you because I need you to understand that prophase, metaphase, anaphase, and telophase are just the cell reproducing. When I say the cell cycle, I could substitute in the life cycle of a cell. Like the entire life of a cell. The life cycle of a cell. And the life cycle of one cell can be broken up into 3 main parts. Interphase, mitosis, and then cytokinesis. Now, of course, there's substages, but 3 big things happen throughout life. Interphase, mitosis, and cytokinesis. And you know that I'm about to expound on all of this. But I'm also going to parallel it to you individually. Okay, so hopefully it'll help this content and these concepts stick. But question, in your life, do you spend more time growing and developing or physically having babies? Growing and developing. Fact. Okay? So, along that same pattern, your cells do the same thing. They spend the majority of their time growing and developing and just a short time reproducing. So this diagram here shows interphase, which is the cell growing and developing. Then mitosis, which you can see is quite shorter. And then cytokinesis, when the cells actually physically split. Interphase is that cell growing and developing. Mitosis is it reproducing. And cytokinesis is it splitting. And then those offspring start that cycle all over again. Now I have that interphase has 3 parts because it does. And I'm going to walk you through each part. The first part is G1. The G stands for gap. G-A-P, gap. So G1 is gap 1. During this phase, the cell grows in size. During this phase, the cell grows in size. This pattern is very much what you did between kindergarten and 8th grade. Kindergarten, 8th grade, you started as this really small 5-year-old, and you finished off as this strapping 14-year-old, or 13-year-old, however old you were when you finished 8th grade. But you grew in size, okay? During G1, the cell just grows in size. S, the S phase, which is the second phase of interphase, or second stage of interphase, stands for synthesis. Synthesis. What does synthesis mean again? To make. During this stage, DNA is replicated. During this stage, DNA is replicated. During the S phase, DNA is replicated. this is you in grades 9 through 12. And what I mean by that is K through 8, if you're a parent, then you know what I'm talking about. And if you're not a parent, but you've experienced this with your parents, which you probably have, between K and 8th grade, you tried a whole lot of different things. Some parents made you play a lot of different sports. Some parents were like, okay, we'll try band and choir and theater and ceramics. Try it all, because you don't know what you're going to like. But by the time you hit high school, you kind of had it figured out, like, I'm really not a basketball player. And I'm actually better at something else. And so you tend to start focusing on things that interest you and that you've been successful at. So we start to see this narrow focus. We've grown and developed, we've just grown in size, and now we've replicated our DNA. Okay, grades 9 through 12. You graduate high school, you have a pretty good idea what you want to do, but what happens in college? How many people finish high school and say, this is what I'm going to become, and they actually become that? Right? Yeah, it's a small percentage, and that's because it's life, and you get on these different pathways, right? So as long as you have the grit and keep going, you're going to make it. You just might not end up the way you thought you would. Like I said, I didn't even go to school to be a teacher. I don't even have a teaching degree. I don't know how I ended up here. I mean, I do know how I ended up here, but nonetheless, you go off to college, which is the equivalent of G2, and that G is gap 2. During this phase or stage of mitosis, the organelles, the rest of the organelles in the cell replicate, and the cell prepares to divide. The rest of the organelles in the cell replicate, and the cell prepares to divide. This is you in college. You found what you really enjoy studying, or hopefully you enjoy studying. You've pursued it. You've become good at it. You have an idea of what you're going to do. And at the end of college, and you've started your career, you are now ready to start a family, according to society. We'll have those of you who don't want to start a family in just a second. But according to society, you go through school, and then you have a kid after you graduate, and you've got a stable career. According to society. I'm going to say that one more time because I was not according to society. Okay? Yeah. I was the 19-year-old with a brand new kid. That was me. So it kind of works its way out. But after you finish G2, the cell believes, and we're going to talk about checkpoints here in a moment, that you're ready to move on and reproduce. So you've gone kindergarten all the way through college, and now you have a career and you're successful or on your way. You are now ready to reproduce. Can you imagine how the world would be different if we all followed this exact same pattern? We would be robots. Someone said yesterday, the line at Chick-fil-A would be much shorter. And I was like, well said. There would be less people, true or false? There sure would be. So, and we'll talk about how that plays into your life. After we're ready to divide, we actually go through mitosis. And mitosis is the actual cell division. Okay, the nucleus divides during mitosis. And I'm going to say that one more time. The nucleus divides during mitosis. Excuse me. In your book, even in your book, in the text, it shows mitosis having 5 phases. Prophase, prometaphase, metaphase, anaphase, and telophase. But then the diagram that they use to teach it to you only has 4. This is very common, because you don't always acknowledge prometaphase. Sometimes it's there, sometimes it's not. It just depends on who's teaching it to you. because I want you to be a boss wherever you are, I'm going to teach it to you. But if nobody ever talks about it, then just know we just went from prophase to metaphase. Like, it's not a big deal. Okay? I just want you to be that one who's just a step ahead of your peers. So mitosis occurs in 5 stages. And then after the nucleus divides, the very last thing we need to do is divide the cytoplasm. Divide the cytoplasm. So cyto tells you cell. What is kine? Movement. So the cell is moving apart. That cytoplasm is about to divide. So the cell grows and develops. The nucleus divides. The cytoplasm divides. I now have 2 new genetically identical daughter cells. And we're going to go into more detail, please note. So I have G1, S, G2. I want to point out this phase right here. These are for all of you who say, I don't want to grow up. I don't want to have kids. There are cells that can make that decision. For example, your neurons, once they formed and matured, they never go through mitosis again. They're what we call amitotic. So they're like, I'm just going to enjoy being an adult and doing my own thing and not having the responsibility of a kid or figuring out how all that works. They're just going to party. That's a benefit. There's a carefree lifestyle. But if a neuron is damaged, what happens? There's no repair. There's no fixing it. Now, if your skin is damaged, it'll repair. You see where I'm going with that? So if the cells decide to hang out over here, and this is what we call G naught, so G sub 0, it's just what we call cellular arrest. They're just hanging out, not doing anything progressive. Okay? So they're just stuck there. Not stuck there, but stuck there. Your neurons formed and developed when you were in utero, so in developing inside your mother, and they will last over 100 years until you die, as long as you take care of yourself. And there's no disease or traumatic injury or anything like that. Okay, questions on the cell cycle so far? All right, so let's go through each part. Interphase. At one point in time, interphase was considered when the cell was resting. But in actuality, the cell during interphase is just like I taught you. It's just going through life. Going from kindergarten to first grade to second grade, all the way to 5th grade, making their little 5th grade graduation. Just doing life, making memories. Interphase. It's broken down into gap 1, synthesis, and gap 2, interphase. On this diagram here, I have these, and these are out of your text as well, because you need to be able to visually recognize what's going on. Okay? So in interphase, during G1, which is K through 8th grade, is my DNA in the shape of chromatin or chromosomes? Chromatin. It's just all loose. It's hanging out. This is you Saturday mornings, eating your donuts. You're like, all right, cool. Right? Until your friends call and say you want to go grab lunch. Then you pull yourself together. Well, some of you do. So you have your chromatin here. You have a solid nuclear envelope. And then after G2, so it skips S phase in this diagram, you can see I have 2 copies of each. Why do I have 2 copies in G2? We went through S phase already where the DNA was replicated. Okay? But I still have my nuclear envelope intact. Here at the top is what that looks like underneath a microscope. Because during lab, you're going to look at it under a microscope. So you can see that you can see the boundary of the cell membrane, and you can see that dark kind of nucleus towards the center. Okay, so interphase. Then we have prophase, which is the first stage of mitosis. The first stage of mitosis. What has my genetic material done now? It's condensed itself into chromosomes. This is when your friends called and said, let's go grab lunch, and you straightened your hair, or you put yourself together, you put a hat on, a hoodie, you look presentable. You're not in your jammas anymore. And what's happening to my nuclear envelope here? It's disintegrating. It's starting to disappear. I also see these structures here that I taught you in chapter 4, where are centrioles. So I see the centrioles there, and I see some lines coming out of them, which I will identify in the next slide even further. But these are basically part of the cytoskeleton. Not basically, they are actually part of the cytoskeleton. They are microtubules, which are a part or a type of filament in a cytoskeleton, and they form what we call spindles. So we call these spindle fibers. So our centrioles are separating, making their way to opposite ends of the cell, and they're starting to spin out their spindle fibers. And I'll talk about the importance of those on the next slide. Oh, again, in prophase, under the microscope, the outside, you can see the parameters of the cell membrane. The nucleus is a little bit more defined. Okay, you can see a darker nuclei there. You can tell something's kind of going on. Just not really sure that it's in a specific stage yet. Prometaphase, which is that segue stage. It may or may not be present when you're doing an activity. So just be open to it not being there if it's not there. But during prometaphase, my centrioles have made their way to opposite ends of the cell, opposite poles. When I taught you centrioles in chapter 4, I told you that centrioles were an extension of the cytoskeleton formed from microtubules, and their function was to direct cell division. So they were an extension of the cytoskeleton formed of microtubules, and they direct cell division. Listen to what I'm about to say here. The only cells that have centrioles are animal cells. The only cells that have centrioles are animal cells. I'm going to come back to that, and you're going to think it's a trick question in a minute. But remember what I just said. Only animal cells have centrioles. So, as these centrioles that are going to direct cell division have moved to the opposite sides of the cell, opposite poles, you can see that their spindles have been thrown out, and some of them are touching a chromosome, and some of them are not. When I first studied this, my biology teacher at A&M, she was not friendly. So I spent a lot of time trying to figure this out on my own, and when I did finally figure it out, I want you to get inside my head real quick because I'm a very simple thinker. I spent forever trying to figure out how do I parallel this to something I understand already. I think of these centrioles as fishermen or fisher women or fisher persons, whatever you want to say that they are. But they're on opposite sides and they're in competition with one another, trying to catch the most fish. So they've cast out multiple lines. Some of those lines are going to catch a fish. Some of them are not. So you can see that these centrioles here, or this fisher person, has connected with those chromosomes. And these ones right here have not connected. I'm going to come back to this slide. I'm going to define those structures and then re-explain what you see so that you understand. What is it called when I have an exact copy of a chromosome? Sister chromatids, or sister chromatids, depending on where you want to put the emphasis. So I have my sister chromatids. Now, normally when people are asked to draw chromosomes, they draw an X. In reality, these chromosomes don't cross. These chromosomes come really close to one another and kind of bend back. They don't touch. That force that holds them together is called the centromere. This is that area where they're held together. You have to know this. It's called the centromere. That force that holds them together. So these sister chromatids, they look like they're crossed over, but in actuality they're not, and they don't even make contact. They're just really, really close to one another. And that force holding them together is called the centromere. Okay? And then on either side of my sister chromatids, I have a structure. It's kind of like a button, but the name of that structure is called the kinetochore. The kinetochore. K-I-N-E-T-O-C-H-O-R-E. Kinetochore. And each of the sister chromatids has a kinetochore on its lateral side, on its outside. And that kinetochore is analogous with the fish's mouth. So if I'm going to catch a chromosome, I got to get its kinetochore. If you're going to catch a fish, you have to hook its mouth. Okay? So here I have my centrioles spitting out a spindle, and that spindle attaches to the kinetochore if it's going to catch that chromosome. That's the attachment point. So the centromere is that force holding it together. The kinetochores are on either lateral side. The kinetochore is the point at which the spindle attaches if it's going to attach. Not all spindles attach. Now, there are two more words I put on this slide so I didn't forget to tell you them. They're not specifically in your notes, but I need you to know the difference because people get confused. I taught you centromere. You know what centrioles are. Where will I find centrioles in a plant cell? Where will I find centrioles in a plant cell?
Oh,
you don't. Because the only cells that have centrioles are neural cells. It's a test question. It's a test question. Now back to my centrioles, which I've only found in animal cells. The term now is centrosome. Centrosome is the area around the centriole. The centrosome is the area around the centriole. You can easily see how people confuse centromere and centrosome, just because the words are really close to one another. Centromere is that force that holds those chromosomes together. Centrosome is the area around the centrioles. So this circle is just showing you like that's where those microtubules are coming from. But that circle along with where the spindles are starting to come from would be the centrosome. Yes, ma'am. Good question. Okay, and where will I find a centrosome in a plant cell? You won't. Why won't you find a centrosome? because you don't have centrioles. I don't know why I had to say it like that. But you don't have centrioles. So, going back, now that you know the terminology, during prometaphase, from the centrioles, spindles are sent out and they connect to the kinetochore of a sister chromatid. If the spindle connects to a kinetochore, we call it a kinetochore spindle. And if it doesn't connect, like right here, these are not connected, we call it a non-kinetochore spindle because it did not attach to a spindle. Oh, no, it is a spindle. It didn't attach to a kinetochore. It didn't attach to a kinetochore. So this is during prometaphase. The fishermen cast out their lines, and if those spindles come into contact with the chromosome, they attach at the kinetochore. They become kinetochore spindles. If they don't, then they become non-kinetochore spindles. So from prometaphase, we then go to metaphase. I remember metaphase middle. And this is a really important stage because what's happened here is each fisherman is starting to reel in their catch. So they're starting to pull, but when you first start to pull in a fish, what does it do? It bites, right? It kind of pushes back. So what we need to happen here is as we're trying to pull them in, they're trying to push back in a way. They line themselves up perfectly on the equator of a cell. So I remember metaphase middle. And why does it have to be perfect? Perfectly straight? Because what's going to happen? They're going to split. And if it's not straight, one side might get more than the other. So this is going back to my 4 and 4 example. I have 4 on this side and 4 on that side. And are these actual X's? No. They're just really close. Okay? So once the fisherman starts to reel in, when this fisherman reels in, only this one's going to come in. And when this fisherman reels in, only that side's going to come in. Does that make sense? So we want them to be lined up perfectly so each new one gets an exact copy. There's no more or less, so we end up with 4. With 4 in each of our new cells. Okay? Oh, and then when you look at this underneath the microscope slide, you can kind of see where those centrioles are with the centrosome, and the chromosomes are lined up. There's a thick line in the middle. Metaphase is probably the easiest one to identify. Then there's anaphase. And for anaphase, I remember apart. Anaphase apart. Anaphase apart. What has physically started happening here is now each of the centrioles, or each of the fishermen, have begun reeling in their chromosomes. So the chromosomes are moving apart from each other to the opposite poles of the cell. Now, are the non-kinetochore spindles moving? Are the non-kinetochore spindles moving? No, because they didn't catch a fish. They're going to have a job in just a moment. But the only ones that are being reeled in are the only ones that are being reeled in are the ones that caught fish, because you're bringing your fish back. Okay? So my kinetochore spindles are being reeled in to pull those chromosomes apart to opposite poles of the cell. Anaphase apart. Now, these here non-kinetochore spindles, they do have a purpose. Ooh, I need to point out this. So they were sister chromatids when they were together because they were an identical copy. As soon as they separate, we now call them daughter chromosomes. They're the exact same thing. It's just how you're referencing it. A good way to think of it is like when you have twins, or you know twins, when you're referencing them, you're like, oh, the twins. But when they're separate, you call them Brooke or Megan. Does that make sense? Like, but when they're together, you're like, oh, the twins. You don't say, oh, Brooke and Megan. You're like, oh, the twins. So they were called the twins when they're together, and so sister chromatids when they're together, and then they become daughter chromosomes when they're apart. So you're just calling them by their name when they're separated. So I wanted to point that out as well. So this is showing you what these spindles are doing. So the kinetochore spindles had lined up, caught a fish, caught a chromosome, attached to a kinetochore, and they were reeling themselves back. They were reeling those chromosomes to opposite poles of the cell. The non-kinetochore spindles, if you read the description, it says they produce a ratcheting action. A ratcheting action. They're going. And when they're doing this, what are they doing to the cell? Pushing it apart. They're pushing that cell apart. So the kinetochore spindles retract and they reel in those chromosomes. The non-kinetochore spindles are pushing the poles further apart. They're extending that cell because it's getting closer and closer to doing what? To separate. Then we have telophase or telophase. I call it telophase, but some people call it telophase. I'm cool with whatever you want to call it as long as you get the right answer on the test. During this phase, what have my chromosomes begun to do? They're going back to being relaxed. So they're no longer chromosomes. They're now considered chromatin. Prophase, metaphase, anaphase, telophase, that was the nucleus dividing. That was the job. So the nucleus is now divided. Look what I started to form again. Another nuclear envelope, something around that genetic material to house it. And during telophase, the cell begins to kind of dent itself in. And that denting itself in is called forming a cleavage furrow. A cleavage furrow. Not like cleavage like as in breasts. Okay? But in a cleavage furrow, what we start to see is something cleaving. Starting to kind of fold in. And of course, when I saw this, I was like, how does that happen? And believe it or not, there's a protein there that takes care of it, but let me tell you what it looks like. Have you ever seen those sweatpants that have the drawstring on them? And when you pull the drawstring, what does the waist of the sweatpants do? It's exactly what happens here. So you pull on that protein, that tubule, it starts to pull, and as it pulls, it's constricting the middle, but it hasn't popped apart yet. Eventually what it's going to do is it's going to constrict, and bounce back. And you're going to have two cells. Like it's going to get so tight, it'll eventually separate. Telophase. I know that this is telophase. The main indication, of course, I have my genetic material, but you can see how I have like a new cell membrane. You can see that it's starting to divide. So mitosis is the nucleus dividing. So we've done that. The last step is cytokinesis, which is the division of the cytoplasm. So I continue to pull that drawstring until it eventually pops. Not pops like explodes, but they pop apart into two separate cells. Do you think plant cells can do all that fanciness?
No.
Why can't a plant cell do that whole cleave and pop? It has a cell wall. It's got a whole cell wall around it that's preventing that practice. So let's look at what it does. I'm going to show you animals first. And this diagram right here is truly what it looks like in your body, just so that you know. But you have it going through telophase. That cleavage furrow begins. You have that contractile ring. Okay? Then it continues to cleave. And then eventually it separates. And you have two new identical daughter cells. This is in animal cells. Again, an indication to me that this is an animal cell is it kind of has a rounded shape. But specifically, there's no cell wall. You can see just by looking at it, it's kind of a rounded shape. Here it is in a plant cell. So they do the whole same thing. Interphase, prophase, metaphase, anaphase, telophase. But they don't go through cytokinesis like the whole popping thing. The cytoplasm is split by the formation of a new cell wall. And until that cell wall is complete, we reference this as a cell plate. So it forms this new cell plate that continues to extend until it's officially formed a new cell wall. Till it's officially formed a new cell wall. Okay, the middle lamella is just what we call it as it starts to deposit. That's the separation. When we talk about, we have spoken about the cell wall of plant cells. I don't know if you remember they had a secondary, a primary cell wall, and they have a middle lamella. The middle lamella is the part that separates the two. There. So overall, the cell cycle includes interphase, mitosis, and cytokinesis. Interphase, then after it's gone through G1, S, and G2, prophase, if there's a pro-metaphase, it will be right here. Metaphase, anaphase, telophase, and then of course cytokinesis. And there's no phase for that because it's just divided in to two new cells. You do need to be able to recognize them underneath a microscope, which is what we're going to do in lab today. So I'll get there in just a moment. Now, all this is really awesome that it goes through this interphase, it grows up, and it reproduces and so on. But we have to have some type of check and balance system. Because what happens if you finish 8th grade, but you're not ready for high school and you still go on to high school? You're setting yourself up for failure. In the state of Texas, if you've been in the state of Texas for any extended period of time, what do we do after 8th grade or during 8th grade to determine if you're ready? You take a state test. Now, we're not going to discuss whether or not we like state tests. I'm just using this for an example, okay? Because I feel you. But at the end of 8th grade, you take this test, and if you score what they consider an acceptable score, they let you move on to 9th grade. But if you don't, what do? You are not yet ready. So you need to stay back in 8th grade. Your cells have the same type of setup. We don't want them to move on if they're not ready. We don't want them to move on if they're not ready. So we have 3 major checkpoints throughout this cell cycle. We have one right here before the cell goes into 8th grade. So it's grown in size. Are you ready to replicate your genetic material yet? And if you're not, why don't you hang back, finish growing up a little bit, and then we'll move forward. And it'll give it some time to correct itself, but what if it doesn't fix itself? What do you think our body does to it? We don't got time to waste. If you're not serious about making me successful, then get out. But we give it a chance. So our first checkpoint is here before we hit the S phase. Our second checkpoint is after we finish college. We got a job. Are you ready to reproduce? Again, what if life was like that? What if you were not allowed to have a kid until you were what society considered ready? Things would be much different. Much different. Okay? But so obviously we don't have a checkpoint. But that's the next cellular checkpoint. Right here. Then if we believe, or if the cell believes that it is ready to reproduce, it will begin mitosis, where it will divide. And there will be one final checkpoint during metaphase. And why do you think it's metaphase that we do the checkpoint, final checkpoint? We want to make sure it's lined up correctly in the middle. We want to make sure it's lined up correctly in the middle. And I've explained all of these here. And a couple things. I do want you to know that if these cells don't meet the criteria for these checkpoints, that they're forced to go back. Or they're destroyed. But these checkpoints are monitored, but I want you to listen to the name of what I'm about to tell you and tell me what you get from it. They're monitored by molecules called cyclin-dependent kinases. Cyclin-dependent kinases. Anything that you can pull from that name. Okay, so movement, because kine means movement. Dependent, so it's depending on cyclin, okay, which makes people think of a cycle. ACEs, what's the ACE? It's an enzyme. Okay, cyclin is a protein, but cyclin-dependent protein kinases. These are enzymes that they're there with their little clipboard when that cell's ready to move on, and it's like, have you met these criteria? And if not, scurry on. And if so, move forward. Go on and be successful. How long do you think that this entire cycle takes for your cells? Seconds? Let's just say on average, it takes about 24 to 48 hours. And why do I want it to take its time? I want to make sure it's going to do a good job. I want to make sure it's going to do a good job. Paralleling to your life, how many of you have really messed up in a class and then you go ask to do extra credit? You go do extra credit and realize that the extra credit is much harder than actually doing the assignment itself. If you would have done it right the first time. I can't even guess how many times my parents said that to me. Well, if you would have slowed down and done it right the first time, we wouldn't be in this predicament. Good point. But we're here now. So, now what? All right. So, we want it to take its time. We want it to slowly go through each of the checkpoints and make sure that the cell is where it needs to be in order to move on to the next stage. Here, I have the CDKs redescribed. These kinases, these cyclin-dependent protein kinases are inside the cell, telling it it's ready and it's not, and so on. In your life, what have you had externally that's kind of guided your pathway? Your parents. So cells have internal controls, those proteins, those enzymes, which are proteins, those CDKs, but they also have external controls, telling them, this is a good time for you to divide. For example, when you were much smaller, your parents did a lot to take care of you. They would make your meals, especially when you were a baby, change your diaper, they would bathe you. But as you grew up, what did they teach you to do? Yes, like here, I still remember the first day I told Isaiah, make your own cereal because I want to go to sleep. And then after, I was like, oh, crap. Because he got upset and went and asked his sister, and to prove a point, she poured the cereal on the floor. But whatever. Okay, I still slept for another 35 minutes before I had to clean up that mess. But I was so excited because I had made him breakfast every single morning, but that morning I really wanted to sleep in, and I knew he could do it. He was ready to make his own cereal. And you know when you teach your kids to make their own cereal, they grab that gallon of milk, and their arms kind of shaking, and you're like, oh. But you kind of just let it happen because once they experience it, they know what it feels like. They're like, okay, now they can make their own cereal. Z makes his own cereal twice a day now. I'm so excited. Okay, but my older daughters, I have them do their own laundry. Z doesn't do his laundry yet because I know he'll break the machine. But the girls, if they want clean laundry, it's not, hey, Mom, can you wash this for me? No. You're 16. Wash your own clothes. So they wash their own clothes. And when they're like, oh, well, that shirt's not clean, I'm like, you better put some body spray on or something because I got to teach you how to cope with smelling bad. Okay? So your parents have those external controls that help guide you and shape you and let you know when you're ready. Your cells have the same thing. When you need to hit a growth spurt, you release a hormone called growth hormone that tells your cells, come on, let's hit it. We have 4 to 6 months that I need you to reproduce at a higher rate. And what happens to you during that growth spurt? You grow. And then that growth hormone starts to secede. And what happens to your growth rate? Kind of slows down. It lets your body get used to it. Now you're this big, let's grow into it. And then a while later you'll hit another growth spurt, and then you'll hit puberty. And this is your body's way of signaling that it's ready for these next stages. So cells have internal controls and external controls that help regulate their cell cycle. What do you think happens if there's no type of regulation? Mutations. I love that you said mutations. Because if there were nobody in your life to make your cereal when you were younger, to make you food when you were younger, would you have turned out the way that you have? No. Even if your food was not fancy, even if it was a piece of toast or whatever, you still had access to those resources. Cancer is uncontrolled mitosis, meaning there's no checkpoints. So there's no checklist that asks you, are you ready for 8th grade or are you ready for 9th grade? We just send you. And then, oh, you're not done with college yet, but you can go ahead and have a kid. These cells reproduce too soon. They don't grow up. Then they have a kid and they're forced to grow up, but their kid is a result of an immature cell, so how does that kid act? Immature. It doesn't know. It's learning from a kid. And that's exactly what cancer is. It's uncontrolled mitosis. The cells have no checkpoints, nobody to help guide them. So they just divide rapidly, no checkpoints. The cell cycle goes from being anywhere from 24 to 48 hours if it's taking its time to just a few hours.
Is it random?
The mutations are usually random, but they can be induced by internal or external factors like UV radiation, exposure to different mutagens, and so on. We have to talk about cancer, and I just happen to, I can do this, but I don't want to get into a deep lecture, but I want you to understand, okay, and I hopefully can get it to make sense to you. But your cells start out nice and healthy. And these are their nuclei. How many cells does it take to start cancer? Just one. One cell has to jack up. One cell makes a mistake. Okay? And then once that one cell has made a mistake, it's going to reproduce faster than the rest. So instead of growing in the straight line that it's supposed to grow in, because they reproduce so fast and they're not mature enough, they have nowhere to go, they just pile on. And that's why tumors grow up and out and not flat. When you have this small tumor, this is what we roughly call stage 1 of cancer. And it will grow, but then it will kind of slow down for a while. The reason why it slows down is because when it was small, it was easy for it to get nutrients. But the bigger you get, what do you require more of? Nutrients. So what doctors normally say, depending on the patient, the situation, all of that, is let's see what your body does. Because our white blood cells have now recognized that this tumor is here and they're trying to attack it. Okay? So let's say I'm the tumor and you're the healthy cells. So I've grown and I've kind of stalled in my growth because I'm low on food. And I'm like, hey, Araceli, can I have some of your food? And she's like, no, you're not even supposed to be here. I'm like, okay. The next day, hey, Araceli, can I have some of your food? No, I told you no the first day. Oh, okay. And the tumor will just keep asking. And sometimes your body fights it the whole time and never feeds it, and then that tumor doesn't move, doesn't change, it doesn't thrive. But all I need is for one of you to share me your resources and say, gosh, I'm so tired of you begging. And then as soon as I get bigger, and then you can't say no to me because I'm so big. And I just start taking it, and I will, once I get those resources, I will start to take off in my growth, but then I'll stall again. This is roughly what we would call stage 2, when I've kind of taken out the resources and started invading the other cellular spaces. And what happens to you when I take your food? Yeah, you starve, so you're eventually going to do what? Expire. Okay? So then I'm in another predicament. Have you ever heard that a fish will grow the size of its tank? Your fish will grow so big if you put it in a fish tank. And if you put it in a bigger tank, that fish will grow. But it will stall when its resources are tapped out. It'll stay that same size. So right now, I'm a fish in this tank. I have to figure out what I'm going to do to get more food, because I've already taken all your food. So I have to start signaling to your body. And your white blood cell count at this point is shooting up. We already know you're sick. Something's not right. The only way I'm going to get more nutrients right now is if I tell your body I belong. I already got the cells to think that, but I need the body to think that. So I'm going to start sending a message that says, hey, I'm here and I'm hungry. I'm here, I'm hungry. And I'm going to keep saying that until someone gets so annoyed at me that they do what? They throw me some food. And food in your body is transported by means of a blood vessel. So when I can trick your body into accepting me, it will give me my own blood vessel. And the process of giving something its own blood vessel is called angiogenesis. Angiogenesis. So when it starts to form this blood vessel, we have roughly made it to stage 3. And this is all just very generally speaking. But now that I have my own blood vessel, tell me what I have. Access to everything. Who will stop me now? Nobody. Nobody. Nobody will stop me. So what will happen to this tumor? It'll grow incredibly large. And then, if, per se, just a chance happening, one of these cells breaks off into my blood vessel, tell me where my blood's going to take it. Everywhere. Everywhere the blood goes. And that cell will get stuck somewhere, and guess what it will form? Another tumor. When they say the cancer has spread, this is what that means. And when it has spread, we roughly say that that's stage 4, and it's starting to create tumors other places. That spreading is called metastasizing or metastasis. You hear people say, well, she had cancer in her lungs, but it spread to her liver. That's how it did that. That's how it did that. And the problem is that because there's no checkpoints, as these cells continue to divide, they get more and more screwed up. So the first one was a mistake, a random mistake, but the more they divide, the more screwed up they become. But do all tumors cause damage? No. A tumor that does cause damage or that is malicious in its intent is considered malignant. A malignant tumor. A tumor that is not malicious in its intent and does not cause harm other than being just a bump is called benign. We reference that as a benign tumor. Okay? A benign tumor doesn't cause aggressive, but it could be like that it's moving something out of the way, or it's causing another process to slow down. Not like it's trying to be malicious, but just the fact that something large is there. Like fibroids, usually on ovaries and such, those are kind of like benign tumors. Like they're just uncomfortable, and you really don't want them there, but they're not maliciously trying to destroy you.
They grow too, right?
They do grow too. Usually, though, they get to be a certain size and they just stop because they don't requisition resources. Yes, ma'am. That's a great question. It is my understanding, and I'm not completely versed in all of this, but I do believe there is a potential for benign tumors at some point, but it wouldn't be the whole tumor. It would be one cell, and so then another tumor. Like it wouldn't be that you had a really nice tumor and all of a sudden it's really meat. Like it would just slowly. I believe that that's a thing. I don't want to speak ill-knowledgeably, but I believe that that's a thing. So here is just a cross-section of liver. And in anatomy, I would tell you that if you usually see white within an organ, that tells you that you've got connective or you've got damaged tissue there. So right here, we want the tissue to be this color. And when you have that, that tells me whatever's there is not functioning. It's damaged. When you take a tumor or a slide, so histology is the study of tissue. When you take it and you put it on a microscope slide so you can see it at a tissue level, you can see both cellular and tissue. Right here, these cells are nice and flat like the way they're supposed to be. But remember how I said tumor cells, they'll stack because they have nowhere else to go. So I know that there's a tumor here because I don't have this nice, even distribution of cells. It's all packed in and stacked. Okay, that's how that would indicate to me that there's a tumor there. It depends on what stage you're in, what kind of tumor, like is it encapsulated or unencapsulated tumor, where it's located. Like there are so many factors that play into, and usually it's just taking care of yourself, and if you can get it removed, like you're in stage 1. And when we remove tumors, we don't just take the tumor. Hopefully it's not this big, but we would take all the stuff around it too. Why would we take the stuff around it? Just in case, I'm bad and you guys are relatively close to me, just so that I make sure that you haven't really taken any of my bad patterns, they'll scoop this whole area out. Just so that it goes on to the cells that I haven't come into contact with yet. So when you remove a tumor, if it's early enough, we take what's around it as well. It just depends on what stage you are, where it's at, and all of that. Because sometimes you can't access the tumors. So, like if it's in the middle of an organ, we're really limited on what we can do. If it's on your brain, specific areas of your brain, we're limited to what we can do. This is a feature story in your text, but we do have stuff like this in Texas, and I just want to tell you the idea, the premise behind it. You have patients who have cancer, and you extract some of their cells. All you have to do is feed them, and those cells will live on, which is the condition that we have here with that feature story. I know here in Dallas, we have cells from a woman's ovarian tumors. She's been dead for years. Her ovaries, a female's ovary is supposed to be about the size of a quarter, really small. When she expired, both of her ovaries were the size of footballs. She was 80, didn't want to undergo any type of treatment, so she just kind of let it take its course. But they extracted some of her cells, her tumor cells, and they're still alive. That tells you that cancer is pretty rogue. Like, it will just go on as long as it has a food source. So because they've extracted her cells, they can test on them and test treatments, and she's not even here anymore. But they can use those treatments and those discoveries to apply them to other patients. And we're starting to see this more and more as we go into this cancer research. And oncology is a very booming science right now. Okay? Now, last topic, which will take me quite short. Only eukaryotic cells go through mitosis. If you are a prokaryote, a.k.a. a bacteria, you go through binary fission. If you are a bacteria, you go through binary fission. So you start with one chromosome. And does anybody remember the shape of a bacterial chromosome? We have linear chromosomes. Bacteria have one circular chromosome. One circular chromosome. They have one. So they replicate their DNA just the same, but it's much less complex because we have a lot of chromosomes and they don't. They have one. And then that genetic material will separate, and the cell will also separate. This process for prokaryotes or bacteria is referred to as binary fission. Binary fission. Main differences between plants and animal cells going through mitosis. What are they? What doesn't have centrioles? Plant cells. The only cells that have centrioles are animal cells. What else is a little bit different? Cell wall and cell membrane. When you're looking at an animal cell, in general, they tend to be a little bit more circular in their shape. When you're looking at a plant cell, they tend to be more robust and like a rectangular square shape. Both go through prophase, metaphase, anaphase. That's all the same. But that cytokinesis, that splitting part, is different because plant cells have a cell wall. When you look at slides today in lab, first of all, are these plant or animal cells? Yeah, it's clearly labeled animal. And their cell membrane is round. These were taken from a white fish blastula. Let me tell you what a blastula is. So when a sperm and egg fuse, we call it a zygote. So that's fertilization, so you have a baby. A blastula is after it's divided a little bit. So what I have here is a fish egg. Okay? Why do you think we're going to use fish eggs to study mitosis? They're growing. They're at the egg stage, they're rapidly growing. So when you look at it underneath the microscope, and it may be dyed a different color, these cells are a roundish shape. You can see like this one is an interphase. It's not really doing anything. But I can tell this is a definite metaphase. Okay, I can see that there's something happening here, but I'm not sure, so I would label that prophase. Here, I got a metaphase. Here, I have anaphase. Let's say an anaphase here. Prophase here. Telophase here for sure. Okay, that's what you're trying to do for the animal cells. You're trying to find them in the different stages of mitosis. You need to be able to recognize them for your lecture exam and your lab exam, and then your lab quiz that you'll have next week. In plant cells, we are going to look at the tip of an onion root. Why the tip of an onion root? That's where it grows. Yep. And mitosis is for growth and division. Make sure you know these models. But this is a model of an onion root tip. Very bottom is what we call a root cap. This part is not growing. This part is to protect the part that's growing. So what are we protecting it for underground? From? What are we protecting it from? And as it grows, what could it do? It might hit like a rock, a line underground, a sewer thing. But this part will hit it first. And this part will immediately communicate to the rest of the root, hey, there's something there, and it will guide it around it. That's how plants grow around things. We call it thigmotrophism. This part is not growing. It's the cap. It's called a root cap. This part here is actively mitotic. This is where the plant is growing, either from the tip of the roots or the tip of the shoot, so the very top of the plant outside. Those are the only places. So when you're looking at your root tip under the microscope, if you look up here, all of these are mature cells, so you're not going to see a lot of mitosis. If you want to see a lot of mitosis, you need to kind of move towards here. And if you see a bunch of like blank cells, that's the cap. You're looking for right here. And you'll see a lot going through mitosis. Like you've got a solid telophase here, metaphase, some prophase, metaphase, anaphase, you can see that there's a lot. If when you focus your microscope you don't see a lot, all you have to do is kind of move that slide a little bit and you'll see some. But I want you to understand what it is we're looking at and why we're looking at them. The very last thing is my summary slide over this whole chapter. For mitosis, mitosis occurs in a somatic eukaryotic cell, and I'll explain somatic on Tuesday. The purpose of mitosis is growth and repair, or the maintenance. The nucleus will divide one time. The chromosomal structure is linear, and there are many for eukaryotes, many linear chromosomes. The product is 2 genetically identical daughter cells. Timing, 24 to 48 hours. We want it to take its time. It can take longer. And if it becomes uncontrolled, it's considered cancer. For a prokaryote, they go through binary fission. The whole purpose is reproduction. It divides one time. Chromosomal structure is circular. 2 genetically identical daughter cells. Timing, anywhere from 30 minutes to 24 hours. Bacteria reproduce fast. But they're very simple also. Why do you think I have mitochondria and chloroplast down here? They used to be bacteria. If you need more mitochondria in your body, guess what process creates it? Binary fission. That is the third and final piece of evidence that we needed to prove that mitochondria and chloroplast were once prokaryotic. The first piece is they have their own genetic material. Second, they have 2 cell membranes. Third, they divide by binary fission. That is the end of this chapter. Any questions?